This is the first of our DNA lectures, and in this one we're just going to cover DNA structure and its process of replication. Just a few learning objectives here. Now, genetic information. What is genetic information? If we were sitting in class, I would make you answer this, but since we're not, I'll tell you that genetic information is the information that is stored in your genome or in your DNA. It's the information that makes you, you. And that's the form that it takes. It takes the form of DNA, and DNA codes for proteins. It has your genes in it, which code for proteins, which kind of create your entire body. Where do you get it? Well, you get it from your parents. You get half your chromosomes from your mother and half your chromosomes from your father. And what is a gene? I've already told you this in class, and I'll repeat it here and a number of other times this semester. A gene is a segment of DNA that codes for a protein. And what is a genome? Well, a genome is your entire set of genes, your full complement of DNA. So genetic information tells cells how to construct a great variety of protein molecules, each with a specific function. It tells them how to make them and when to make them and in what quantity. DNA is made of DNA is a nucleic acid. It is made of nucleotides. And we did this a little bit a few weeks ago. Nucleotides consist of a 5-carbon sugar, which can be ribose or deoxyribose, a phosphate group, and one of several nitrogenous bases. The phosphates and sugars link together to form what is known as the backbone of the DNA. So the backbone is formed by the phosphates and sugars. DNA is 2 polynucleotide, so multiple nucleotide chains that are joined by hydrogen bonds between the nitrogenous bases. So hydrogen bonds are the bonds that form weakly between polar molecules, and that's what's happening here. So the nitrogenous bases link together and form kind of this ladder pattern, with the rungs being the bases and the rails being the phosphate sugar backbone. These strands are organized in anti-parallel conformation. That basically means, and you can see it here, that one points up and one points down. The bases that are part of the nucleotides come in 4 different kinds. 2 overarching categories, the purines and the pyrimidines. The purines are adenine and guanine, which we will know as A and G, and the pyrimidines are thymine and cytosine, which we know as T and C. The most important rule for the structure of DNA is that A will always bind to T and C will always bind to G. ATCG. So when you're figuring out which strand of DNA to match up to the original strand, you look at the bases and know that A and T match and C and G match, and I'll show you that. These are called complementary base pairs. So the bases are A, G, C, T, and the complementary base pairs are A to T and C to G. So look at this pretty structure. You see 2 polynucleotide chains are antiparallel and held together by hydrogen bonds between the complementary base pairs. And then the whole thing is twisted into this lovely double helix shape. So here are the phosphate sugar rails, right, the blue lines. You can see phosphate sugars linked together, one of them pointing up and one of them pointing down because they're antiparallel. And then the bases. So C to G, A to T, G to C, C to G, T to A. All the way down the strands, the base pairs match up like that, and they hold the 2 strands together. Now a gene, again, a portion of DNA that contains the genetic information for making a particular protein. A gene is a segment of DNA that codes for a protein. So genes are widely variable. A single gene may be thousands or even millions of base pairs long, while the human genome itself, the entire complement, is 3.2 billion DNA bases long. So a single gene can be a pretty big chunk of DNA. The term gene expression, which you may have heard, is which proteins are produced in a particular cell under a particular set of circumstances. So lots of things happen inside cells to make genes be expressed or to produce their particular protein. If you take some kind of drug, I always relate this back to drugs because that's what I've studied for so long, that affects your brain in a certain way, you may end up turning the protein dopamine off in your brain. So, your genes stop making dopamine receptors. That's kind of a vague example, but that's what happens. It responds to drugs, you respond to hormones. Your genes respond to the conditions of your body, and gene expression is how the proteins are produced in those circumstances. So in the nucleus, DNA is wound around these proteins called histones, okay? Histones will move on and off the genome as the parts are needed to be expressed. So this is chromatin, right? The unwound DNA. This is what the DNA looks like in interphase, all unwound. And then this is, they show you down here, a metaphase chromosome, what they look like all wound up. So histones bind to this DNA, but then they come off when the DNA is read in order for the genes to be accessed and the genes to be expressed. So let's talk about DNA replication. Why does DNA need to replicate? When does DNA replicate is another good question. So DNA needs to replicate before cell division because remember, after mitosis is complete, you have two identical copies of the same cell. So you need to double the DNA in order to be able to separate it into two identical copies. When does that occur? Well, if you remember, that occurs during S phase of interphase after the cell has decided that it's ready to divide. So here's what DNA replication looks like. The first thing that happens is the strands of DNA begin to separate. So the hydrogen bonds between the two strands break apart and it unwinds. So that exposes the base pairs in the middle. There's an enzyme called DNA polymerase. So DNA, poly, multiple, and ase means an enzyme. So it's making the polymer of DNA, adds the nucleotides to the unwound strands, and stitches together a new sugar phosphate backbone. So that's what's happening down here. The strand's unwinding, and there's the free nucleotides coming in and joining with the unwound strands and getting their new backbones stitched together. So DNA polymerase is the enzyme that facilitates this process. As a result, you end up with two complete and identical DNA molecules. So look down here, you can see the dark blue is the strand that's come apart. Okay, that's what they're showing you. That's the dark blue, that's the original strand. So they've pulled apart, and the DNA polymerase is adding these light blue free nucleotides onto the other end. So this original strand had ACAT, this one had TGTA. Those were the complementary base pair sets, and so they just add the one that corresponds. So this one adds T to A, G to C, T to A to T, and this one adds A to T, C to G, A to T, T to A. And what you see is that these two now are completely identical. So the DNA is perfectly replicated. The genetic code is the correspondence between a DNA triplet and an amino acid. So what is a DNA triplet? It simply is three bases. A DNA triplet is like A, C, T, or GGG, or GCG, or ATG. Any three bases together is a DNA triplet. Every DNA triplet relates to one amino acid. And when we do translation, you'll get to see how the amino acids line up based on what the DNA triplet code is. So one DNA triplet to every amino acid. That is called the genetic code, and it's how your genes make proteins. Other sequences in the DNA code for the beginning or the ending of the synthesis of the protein molecule. So in which organelle is the genetic code stored? That would be the nucleus. And where does protein synthesis occur? That is in the ribosome. The ribosome is out in the cytoplasm, not in the nucleus. We'll talk a lot more about this in the next lecture on transcription and translation. The last nucleic acid I want to talk about is RNA. So RNA is similar to DNA, except its major two differences are that it is single-stranded and that it has a U instead of a T. So U matches up with A instead of T with A. So anywhere that you would have a T normally, now you're going to have a U. It is made of ribose sugar instead of deoxyribose. That's why it's RNA instead of DNA. And it can also be found in the cytoplasm, while DNA resides primarily in the nucleus, only in the nucleus. RNA can go back and forth. RNA and mRNA and tRNA, those are two subtypes of RNA, are absolutely critical for transcription and translation. So we'll talk about them more in the next lecture. So that was DNA structure and DNA replication, and a little bit about the genetic code and gene expression. We're going to use all of this knowledge from this lecture to go and talk about transcription and translation in the next one, which will really build on this.